Yeah, so let's get started. So this is the final day of the masterclass. Thanks for accommodating the adjusted time, for the transit strike and all that. So far we talked about just a recap. We talked about complex spatial networks and sort of different ways to characterize them, traditional metrics from network science, and how good or not so good they are, and then some examples of more purpose-built metrics. Day one. Day two, we talked about sort of distribution of flow networks in biological and physical settings, complex spatial, versions of those things. Adaptation, we talked about the Bryce paradox a little bit. We talked about some different sort of modeling approaches. So we sort of deeper dive into flow networks. And now today we're gonna change gears a little bit and go to sort of shape outcomes in biological systems, so morphogenesis, but with the mechanical network sort of spin to it. So if you remember this plot, I've showed this plot all 3 days, just as the sort of overall framework, right? So the first 2 days we we started, down here, way out on the topological complexity axis and tried to push from there up into the middle. And now today, that was this. I'm kind of trying to do this in the last 2 days. And now today we're going to kind of do the opposite thing. We're going to start sort of more obviously geometrically complex system and think about ways to sort of push in to add network ideas, to add topological ideas, and push into the middle that way. And so, just let's see a little list. Okay, and so how are we going to do that? What are the sort of the details of the approach? So our story, our play needs players. And so we're going to concentrate, there's lots of different important shape outcomes in biology, many different kinds of tissues, many different organ precursors. They're not all epithelial, so we're going to concentrate on the epithelial tissue. Who in here knows what epithelia is? Okay, Florian, good, thank you. So epithelia is a specific kind of tissue where you have a layer of cells, right? So these are cells, and they have very tight mechanical connections to one another. So this is a cross section, right? These blue little lines connecting them is you have this sort of junctional network. You have tight junctions. You have a bunch of different junctional machinery made out of proteins and protein complexes that form these sort of bands connecting all the apical cells. And in many apical tissues, if you kind of look down on them from above, right, they look like a 2D cyclic network, right? They look like a tiling, right, which is like a 2D cyclic network. And, there's a lot of active elements inside the cell. The cytoskeleton is sort of filled with mechanical motors, sort of usually actin networks with myosin motors. This is another network component, right? So you have this internal network mechanics going on with the actin networks and the way myosin is sort of, transducing stresses and strains across that network. And then on the basal side, so I should say that one of the properties of these sheets, these epithelial sheets of tissue, is that the two sides of the sheets are distinct, right? So the one side, which is often thought of as the top side in some wishy-washy way, really the apical side it's referred to. So that's the side where these mechanical connections tend to be sort of closer, between each other. That's often the side that can be imaged better because there's lots of ways to image these connecting networks. That's one side. The other side is called the basal side, like for the base of the cell. And there's lots and lots of extracellular matrix is secreted, and then some mechanical connections between the cells and the matrix. So that again is another complex network, actually, on the sort of subcellular scale. So there's lots of different networks mechanical networks interacting. And there's very interesting questions about the mechanics of these subcellular networks in the cytoskeleton of the ECM. We're not going to talk about those today so much, but other than the fact that we're going to sort of implicitly integrate over their effects to get some sort of larger scale sort of emergent stresses and strains that then we think about in the context of the cell network, right? The overall network you look at by looking at the cells and their neighbors and their connections. Yes.
And can you give a couple of examples of the epithelial tissue?
I can indeed. So many precursor tissues in the body in animals are epithelial, right? So neural tube is epithelial. The precursor to the brain is epithelial. The heart is epithelial, like so. This muscle tissue is this neural tube. This is the heart. You know, the precursor to the heart is epithelial. Your gut lining is epithelial. Anything that sort of forms a barrier in the mature case is epithelial. So like your skin is epithelial. Your all the cells lining your gut are epithelial. And then many organ precursors, even if the later scale, later time organ is no longer, just a barrier, like a 2D barrier, the early stages of that organ are often an epithelial sheet that then makes some complicated shape and folds up into some much more complicated scenario. It has other cell types. Then you get you can get epithelial mesenchymal transitions and things later times to sort of fill in different kinds of cell types. But in many cases, the liver, the pancreas, fly wings, limb buds, all epithelial at early times.
So are we talking about the sheet that is around the tissues then?
No, we're talking about the whole tissue at early times. There's nothing but that. And there'll be more examples. You'll see more later. But at very early times, the organ precursors are often just a sheet of cells that are connected. And then they bud up, and then more cells come, inside, and those the shaped things can also get contorted and fill in other ways with itself. The liver, in fact, is a special case of the sheets becoming so contorted that the whole volumetric tissue is still epithelial. It's just epithelial in a way that like you have the sheet that like folded up and contorted in so much that it's filling space essentially. Okay. Any other questions?
But as epithelial, I mean, it's a sheet.
It's a sheet. You think of
a sheet of cells, so often it's on the outside of things, right? And then also it's folded up in the liver.
But the liver is a special case that creates.
I guess the first approximation you can think about, like a surface.
Yeah,
kind of.
Like the surface of a lumen. Well, if you have a liquid inside with a sphere of tissue around it, then that sphere of tissue around it is epithelial.
Yeah.
So that's another thing you see a lot. Cysts and things like that are all epithelial. Okay. And so so these are some examples of epithelial versus in development. And so how do people think about what are the classic ways that people think about how these shape transitions happen, right? So you have it's a sheet or like a sheet that maybe closes into a tube, but then you get much more complicated shape transitions. What are the ways that people think about the mechanics of these shape transitions? And so far, most people think about, you have this sheet, and then you can sort of make the junctional top, the top of the cells want to shrink, and the bottom of the cells, the basal side of the cells maybe wants to grow or stays the same size. And that can lead to folding, right? Or creation of like cup-like shapes, right? So this sort of cell apical basal aspect ratio mechanics is very, very classic. Like this is probably the dominant mechanism that people think about typically in morphogenesis. And this is an example of gastrulation. It's sort of a zoom in of gastrulation of the fly. So this is when you have the fly embryo at very early times is just an epithelial ellipsoid, essentially, with no other features. And the first sort of feature that appears is this gastrulation, this sort of infolding of what will eventually then become the internal cavity of the fly. And so this is the creation of a fold, and people think about this as being driven largely through these changes in cell aspect ratios. Okay, and then if anybody ever thinks about anything else, it's usually thinking about instabilities. So you have mechanical instabilities that can occur, usually being driven by some building up some stress in the tissue, or especially from interactions with surrounding tissues or with the secreted ECM, things like this. So one of the classic examples of that is this sort of curling of the gut mesentery. This is from chicken, I think, if I remember correctly, where you have one tissue and then another tissue, and they're connected by tons of ECM. So it's almost like an elastic sheet. And then one tissue just keeps growing and growing and growing, and the other one is static, and the ECM is static. And so you get compressional instability on the one that's trying to grow, and so it buckles and curls up over and over again in the sort of stereotypic way from a buckling instability. Okay, there's really nothing more than like your curtains, right? If you have curtains and they're taut at the top and they're kind of like curly back and forth at the bottom, that's because there's just more material on the bottom than there is at the top. And you make it, tight at the top, and they have to buckle and curl up at the bottom. Right? There's nothing mystical really going on with it, but it's a nice way to explain some shape development in markings. Okay, and so, but there are other ways to create shapes, right? So, for example, in sort of engineered systems, so let's say, for example, soft robotics, or sort of this generation of like, tunable lenses, or these sort of funny folding devices, right? There's lots of like cool, different, crazy ways, right? That you can make things, drive shape changes that aren't necessarily, mapping cleanly or even at all onto those two biological mechanisms we talked about. So one might wonder, is there actually some more, space in the sort of biological mechanistic design of shape transitions that has not actually been so well explored? Right. And notice, of course, that we have not talked about mechanical networks yet. The first two things had nothing to do with anything in the plane of the sheet, right? Like the network topology or the mechanics working out over the network in the plane of the sheet is not addressed at all by the first two biological things. And so, in that respect, one can think about shape programmability or programming shape change again, more from an engineering point of view. In different ways, right? Of course, everybody knows about origami, right? So origami is about, making a certain series of folds, right? Folds are sort of just pure extrinsic changes in curvature to generate a shape from a flat sheet. You can do kirigami, right? Which is a generalization of origami where you're allowed to make cuts and folds, right? And so then you can generate, so the thing about origami is because it's only folds is that you can't change the metric curvature of your surface, at least locally. You can create sort of fake metric changes by folding the paper sort of over onto itself to create like what appear to be effective metric changes, but they're not really there. In kirigami, you can actually create metric changes because you can hide all the metric changes, in the holes that you cut. And then, the effective surface will have metric changes, but, the sheets of paper themselves will always be flat, metrically flat sheets. Does everybody know what I mean by metrically flat? So what I mean by that is that the local distances in the sheet of paper don't change, right? So if you have a sheet of paper, right? I have a sheet of paper, I can bend it, right? I can give it a curvature, I can give it a geometry. But if you look at the distance, but the distances of the arcs and the letters that I've written on the sheet, none of those are changing as I'm doing this, right? So the local metric is constant, right? This is if you have ever watched any like cosmology or astrophysics, popular science videos where people talk about the curvature of space-time and how like a little ant could like not tell the difference between a cylinder, right, and a flat space because locally all the distances are the same. It's the same thing, right? So, any kind of surface you can get by like just simple contortions of a piece of paper all have a flat metric. So it's the same distances as a perfectly flat sheet of paper. But there are many shapes that are not a flat metric, right? You can't take wrapping paper, right, and smoothly take your flat wrapping paper and smoothly wrap a basketball, right? Because a sphere does not have a flat metric. Likewise, if you have a vase, right, that has a high negative Gaussian curvature, you also can't take your flat sheet and smoothly wrap that. So kirigami gives you a way to sort of effectively generate non-flat curvature, but again, for these as models of biology, these are obviously no good, right? Biology is not, there's not, you don't generate shape by the mother of the organism, like, folding it into the right shape, right? You don't generate shape by cutting a whole ton of holes, right? So these aren't great. You can have multi-material lattices. This is getting better. But then I think the really appealing things from the shape programming point of view, in my opinion, are things like hydrogels, where you can have gradients of local isotropic swelling, right? And if you're clever about the way that you sort of lay out those gradients of local isotropic swelling, I'll give examples in a second, you can generate, interesting shapes. or something called a liquid crystal elastomer or a liquid crystal solid. Somebody know what that is? One of my guesses no, maybe for you. So a liquid crystal solid is a rubber system or a polymer glass if it's overly cross-linked, where you have a bunch of sort of, let's say, nematic or other liquid crystal phase forming molecules in the matrix of the rubber. And they become coupled, the order parameter of that liquid crystal becomes coupled to the local shape of the rubber. And that's a bunch of physics speak for saying how aligned these, like you have a chain of rods instead of beads, and then how locally aligned those rods are, make it so that, you have your polymer will like be super, elongated if they're all lined up, and if they're all random, then you just have a random walk again, and it's not elongated in any one direction. So you can generate length changes sort of along the alignment direction. And also, sort of a Poisson effect, like you get contractions, because if you were isotropic enough, suddenly you're all lined up, then this direction moves in, right, and that direction moves out, right? So it's anisotropic. Okay. And so now in those last two cases, one can think about something called the spontaneous strain, right? So like if the material changes its local properties, to create a shape, we call that a spontaneous strain. So you can create a shape through external forces, right? So here's the, the sculptor, you're constantly making external forces onto the creature, right, to make this transformation of a man into a werewolf, right? And so the idea is that like locally, you're putting external forces, you're stretching and, changing the material, externally. And then, in a world in which this could actually happen, where you don't need a sculptor to do it from the outside, and the material sort of changes its length on its own because shifts in its own ground state, that would be a spontaneous strain, and it would look like this, right? You would have, of course, the intended animation of the werewolf transition. And so the idea here is that you're not putting stresses into the system from the outside, you're changing the natural lengths of springs or the relationships in the system from the inside, right? And of course, in some sense, these are dual to each other, right? But it's very valuable to be able to think about it in this way sometimes instead of only in this way. Okay, and so I give this example of the werewolf transition, but like, can you actually see? Can I convince you that you can get shape, simple shape, in a way that you understand it from these spontaneous strains? And so again, let's imagine something like this uniaxial anisotropic thing, like the nematic solids. You can abstract that away to thinking about, you have a isotropic box, and then upon some change in the material state, so like it's the temperature changes or the light is shown on it or something, then it extends one way and it contracts the other way. It contracts by an amount lambda less than 1, and it extends by an amount lambda to the minus nu, which is greater than 1. Okay, and then for those of you that know a little bit about epithelial tissues, there's this concept in biology called convergent extension, where you have neighbor rearrangements in the tissue that drive a tissue patch to go from one shape to an elongated shape. And again, like when people observe this, they only think about it in 2D. They only think about like this one patch changing shape. They don't think about making some kind of pattern. But if you were to make a pattern, right, let's introduce a topological defect. And so now topology starts creeping in, right? So let's introduce a topological defect to the spontaneous strain field, right? And so the idea is that along all the dotted lines, we're gonna, this is the alignment. And so along all the dotted lines, we're gonna shrink. And perpendicular to all the lines, we're gonna extend, okay? So we shrink by about lambda, so the perimeter of a circle here goes from its starting p to lambda times p, which is smaller than p. And a radius, at the same time, right, goes from its starting r to lambda to the minus nu times r, which is greater than r, right? And so what that means is that you now have a relationship between the perimeter and the radius that is not p equals 2 pi times r, right? So that means that you've broken Euclidean geometry, on this surface. And in fact, you can work out what the new relationship is. It has this lambda to the minus 1 minus nu. And because I never told you which circle I picked, this is the same relationship for every circle, right? It's a self-similar system that's scale free, and so it has to be a cone. It's the only shape that has that property. And you can actually solve it, I didn't put it on the slide, but you can actually solve for the opening angle of the cone as a function of the magnitude of lambda, which tells you how much. It's an easy, algebraic solution. So this is for, a simple example, sort of motivating that, you can have these spontaneous strains can drive, some simple understandable shape changes. It's actually very general. If you write down what's called the spontaneous deformation gradient tensor, blah, 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 which is really just, encoding what the pattern is and how much it's changing, what the metric is doing locally. In fact, really is directly related to the metric, like that object, the deformation gradient tensor squared, which is just these length changes kind of encoded everywhere, is just equal to a target metric, right? So you can put in a target, you can design a target metric for your system, by putting in some pattern, and then drive that shape change to that target metric. And so it really is changing the rest lengths of the springs. You're changing the ground state, the elastic ground state of the system to this new target metric. So this, of course, these tensors are smoothly defined. So these typically, it's difficult to work with them with topological defects. With topological defects, something else, but that's the detail that we don't need to get into. But it works in both ways. Okay. Questions so far? And this is kind of a lot. Yeah.
So why is it called instantaneous again?
So the spontaneous, the choice of the word spontaneous, it's a little historical. The idea is that it means that it's to differentiate from a strain that arises from sort of external sources. It's that the material is doing it itself. So it's spontaneously changing its local lengths.
So do you mean that such a material exists where you can sort of make a cone out of this?
Yeah.
It exists.
It does exist, yes. I don't remember if I have a slide showing.
How is it induced then? By temperature or?
So it couples super strongly to the magnitude of the liquid crystal order. So temperature, the first versions of this were all done with temperature, and then some very, very clever chemists came up with ways to dope light responsive molecules.
Okay, and
then
alignment.
And so you can do it with light. You can also do something very similar with
Gels.
with humidity gradients, with the hydration and gels. So there's lots of different knobs that you can turn to drive the internal state changes. Temperature was the first and sort of the easiest to see how it would work, but there's also now there's light, there's humidity gradients, there's what was the other one I said? Others, So there's you can do it, I think in some cases with electricity, that's a little harder.
Piezoelectricity.
Sorry?
Piezoelectricity.
Yeah, so that's a little different to this, but it's similar. Yeah, it's in the same ballpark. Okay. And so so could biology maybe be doing something like this, right? So maybe there are morphogenetic processes that drive a shape, like this instead of one of these other ways. And I should say, by the way, that there are lots of morphogenetic transitions that are not understood how the shape appears, because those two classic ways of thinking about it are really insufficient. So this is not there is a real question that needs to be answered here. Biology is in need of new mechanisms or new ideas for ways to explain shape changes. And we'll come to one of those examples later. And so indeed, right, there's actually a whole bunch of different things. You could have cell divisions that are always oriented one direction. You could have, the cell you could elongate, in some pattern. Yeah, and you could also have these sort of neighbor events. There's lots of cell behaviors that occur that have mechanics in the plane of the tissue that would lead to either isotropic or anisotropic spontaneous strains in some coarse grain sense. And we'll talk about that coarse grain in a minute also. Okay, and so the roadmap for the lecture today is to give, just kind of step back a little bit from the network stuff and really talk about just simple spontaneous strains in a kind of a warm-up, like a 1D warm-up setting that actually ends up being an interesting biology problem. And then I'm going to jump to the board and explain to you what a vertex model is, and give you some theory about this coupling between spontaneous strains in cell behaviors and vertex model. And then I'll give this example of the early formation of the flywing, and how we think that is the sort of first real prime example of this sort of metric shape programming, in plane cell behavior. shape outcome through a mechanical network. Okay, that's the roadmap. So we're going to start with cephalic furrow. Okay, who here knows what the cephalic furrow is? Probably nobody. Florian knows it. Very
I
good.
mean, you told me.
Okay. You cheated. I told you.
Yeah.
Okay, so the cephalic furrow. So this happens during gastrulation, which I showed you like this close up from underneath. The movie I showed before was like from under there. Oops.
Maybe you should quickly say what we see here.
Yeah.
Because I think it's not obvious for everyone.
Right. So yeah, good point. So this is the fly embryo, right? So there's an eggshell. Which is going to be important in a minute. So there's a hard eggshell around it. These little lines are the junctions between the epithelial, the apical sides of the epithelial tissue. And then I'll play this a couple times because there's actually a lot going on here.
So these are the cells.
Yeah, so these are the cells. So there's lots of cells, right? And this scale bar, what is that scale bar? That scale bar is probably 100, maybe 50 microns. Actually, not sure. It's either 50 or 100 microns. Judging from the size of the cells, it should be, it's probably 100. So this whole thing is like
half a millimeter
to a millimeter big, right? So it's a fly egg. And so this is just really, it's just a sheet, right? This is not a filled ball of cells. It's a sheet with yolk on the inside. Okay, so the tissue itself is just the outside, and then you get all these crazy flows. The tissue moves all around. So it's going to here, now it's rewinding, right? So it's just playing again and again, but this is forward time, that's backwards time. And this is forward time. Okay. So notice that like you get a bunch of flows going down initially, and so those are the flows that are sort of helping drive the gastrulation invagination. So this is the ventral furrow, if you've ever heard of that. At the same time as these tissues are flowing down, you get a bunch of tissues flowing up and around. And there's this little divot you can follow that moves across the top of the image. And then the cephalic furrow is this fold that happens here, right? And this is the postdoc from Pavel Tomaczak's lab, Bruno Lupini, who did a lot of the experimental work on this. The cephalic furrow is interesting because nobody knows what it's for, right? So for example, the gastrulation, the ventral furrow, really forms the entire internal cavity of the organism, right? Like that is critical. You need that, right? The cephalic furrow, on the other hand, it forms very stereotypically, very repeatedly, and then after a couple hours, it just unfolds. And all of the cells that participated in the formation of the cephalic furrow don't do anything different. They don't have a different gene expression profile. They don't have different sulfates later. They're not part of a different, organ or suborgan or sensory organ or anything. Nothing appears to be different. They do nothing. Nothing is different. And so nobody knows what it's for. Why does it do this? So maybe it's an accident, but actually there are special sulfates, cell activities that drive the formation of the cephalic furrow, right? So you have, this is this cross section, and you have these special cells that like shrink their lateral side. So now this is a cross section of the sheet. So this is the apical side is up here, and so they sort of shrink down, and that causes the other cells to kind of get pulled along with them, right? So they initiate this formation of the furrow. And so then you pull down, the whole thing kind of pulls down into this fold. Okay, so there's some actual sort of programmed activity going on here to drive it, and that programmed activity is under shockingly fine genetic control. So the way this works is that there's two different genes being expressed on the tissue, even skipped and buttonhead, so in purple and yellow, and the even skipped mostly has to do with the formation of patterns, later patterns on the body of the fly. And so they're sort of, this is the head side over here, this is the body side over here. And buttonhead has mostly to do with, this patch up here that has to do with one of the organs later. But it turns out that there's precisely one line of cells, of one cell, like one cell thick line of cells, where buttonhead and even skipped overlap, right? So there's one line of cells that is literally one cell thick that express both of these genes. And that line of cells, having both of those genes being expressed, that is what tells them that you are the initiator cells for the spot furrow, right? So it is really like delicately patterned to be a very specific line of cells that does this initiation, okay? So, you know, it's sort of in some sense genetically important for the fly apparently to do this, and yet we still don't know why it's doing it. And so, yeah, so again, this is just what I was just saying, you have this very specific, you know, gene expression that leads to the mechanics of driving the thing. Okay, so what could the function be? So here again is the little cartoon of the fly embryo. You get the ventral furrow forming sort of underneath. What else is going on? Well, there's also that little divot I said before. This is called the midgut invagination. That's also important for later later actual anatomical feature. You have the extending germ band. That's this sort of flow pushing things all the way around this way. And then there's this falc furrow, right? So, you know, another way to think about it is the ventral furrow forms the mesoderm, the midgut invagination forms the endoderm, this falc furrow appears to form nothing. Okay, but you know, so what Bruno did is he made different kinds of mutants, right? So if the initiator cells initiate because they are expressing, you know, they're the only cells expressing both of those two different genes, then if you mutate, if you mess up individually those genes, then maybe you can mess up the formation of this falc furrow, right? And so indeed, if you do, this is knocking out the button head ones, the one on the head side, then the furrow does not form. Right, so this is the wild type, so we have a nice big clean furrow in the correct location. Here's the button head, there's no big furrow. But notice, there's something happening, right? Like, there's these sort of smaller folds in a bunch of different locations kind of appearing. These ectopic folds pop in, right? So maybe this is a clue, right? So maybe, right, maybe there's ectopic folds are doing something. And so actually, Bruno looked more carefully and realized that there's these mitotic domains, right? So the way that the cells proliferate, they don't just all proliferate at once. There's sort of patches of cells that proliferate in an order on the head side, especially. So you have like this patch goes, and this patch goes, and this patch goes. And these different mitotic domains are sort of these circles here, and the ectopic folds that pop up, like on any given experimental run, you don't get all of the in-betweens, but you get, you know, some of them, right? So this is indicating that, like, okay, maybe the expanding mitotic domains are somehow driving, you know, maybe they're driving these folds. Maybe you're getting that. Maybe this is a buckling instability, right? Here's another example of, you know, zoom in the same thing. This is the light, the color here. The lighter the color, the more it's expressing genes associated with mitosis, and then the fold is appearing right in between these two cells, right? And so maybe the hypothesis was, okay, maybe these ectopic folds are coming from, you know, the sort of stress buildup from the expanding mitotic domains. And remember, at the same time, the germ band is pushing from over here. All this tissue on top is flowing towards this region, right? So there's the recipe, the ingredients are here for maybe sort of a compressive overload.
This is still in the shell, right?
In the shell, and that's
Because
also
it's pressing against.
Yeah, so it can't buckle out.
Exactly.
Right? It
So
can't
it
get
has
bigger.
to.
So it would have to go in. Yeah.
But there's a fluid inside. I mean, there's also.
Yeah, there's yolk inside.
Yeah.
But that's presumably why, you know, when they do fold in, they fold in really tightly. And the cells can also exchange fluid with the yolk as well. Right? And so, yeah, so we made a simple sort of 1D elastica model with a spontaneous strain to sort of capture the mitotic domains and the growing cephalic furrow. The idea is now that you have like a very simple network in the sense that it's just balls and springs in a line along the shell, right? So we have the vitelline envelope, which is the shell. We have our elastica. We push in on one side, we grow into certain locations here, and then to turn on or off the cephalic furrow, we have an intrinsic curvature associated with it. And we can write down an energy functional for the whole thing. right, where we have a stretching energy term and a bending energy term, and we have the rest lengths, right, and the spontaneous curvatures, and we can change the natural rest lengths to create spontaneous strains. Now, again, this is highly simplified because it's only 1D, right? So we're not moving to the full network picture yet. But
Wait, sorry.
Yeah.
Maybe it's a stupid question.
well,
So 1D is because you consider a line,
a line, yeah.
but this line lives in a 2D surface.
Yes,
Because
exactly. So
otherwise you don't have
it's quasi 1D.
Curvature.
That's right, that's right. So it's 1D in the sense that it's
That your object is 1D.
the object is a
because it's
1D line, but it's living, but it can, you know, it has a
Go up.
2D.
Yeah, exactly.
It has a 2D degrees of freedom.
Yeah.
Yeah, so it's better, maybe better to put, say quasi 1D, 2D, that's right, yeah. That's a good clarification, it's important. Okay, and so there's energy functional, there's really only one, everything we can get from the measurements of the experiment, except we don't know the stiffnesses of the tissue, right? So there's this dimensionless bending rigidity, which is scaled against the stretching rigidity. And that's the only thing we have to fit, right? But
You
don't know anything else.
You discretize here, and this is a certain choice you make. The number of discretization,
Yeah,
or
that's
you
right.
do cell junctions. Or
No. So the idea here is this is coarse grained over the cells. So
Okay.
it's a choice. And we tried different amounts of
Okay.
discretization to make sure that it wasn't sensitive to that. But you're right. The numerical discretization here is yeah.
And so bringing it back to a network, you have like a one-dimensional
Exactly.
network.
Yeah,
grid.
it's a one-dimensional network, which you know, in principle might not be that interesting, but in this case actually is, it does the job. So I should say, by the way, so the theory, this was with students from my group, Abhijit and Alicia, who've both moved on now.
And
Yeah.
how do you incorporate flow?
Flow?
Yes.
We don't.
At the moment.
Yeah, not yet. Yeah, there's no need to incorporate flow yet. So we incorporate the sort of the germ band sort of pushing that material pushing in just by we're modeling essentially this part of the tissue, and then we change and they all have the same rest length, and then we have a boundary that we sort of keep moving in, right? We just literally are pushing in.
Okay.
Right. So the flow of the germ band is occurring from just moving that boundary. Okay, and so we can now sweep over the one sort of parameter that we need to fit and see what we get, right? And so as unsurprisingly, right, as the bending rigidity gets stronger relative to the stretching rigidity, you start, you know, at sort of very cheap bend, you get lots and lots of folds, right? You put, you know, in whatever state you happen to be looking at, you fold all over the place. And as you increase the bending rigidity, you get fewer folds, and then you get no folds at all, right? You destroy the instability, unsurprisingly, right? And so then you can play around with, in the model of having, you know, germ band extension and mitotic domains, or just each individually, right? And what we know, right, from the biology is that, well, actually, I haven't shown this yet, but what I'm going to show in a second is that, you know, we can, if there's a significant difference in the actual biological behavior between having both of these things, and this is with no cephalic furrow, this is with cephalic furrow knocked out, right? No cephalic furrow, only looking at ectopic folds, you can have both of these things, or you have only the germ band pushing, or only the mitotic domains. And so you can go back to the biology and try to do a very, very difficult thing, which is make a double mutant in this case. So Bruno did, you know, some really amazing genetic work, and also Marina, this is a student from Pavel's lab that did part of this with just laser cauterization. So you can keep the germ band from extending by burning it onto the shell. And if you do that, right, so this is the cauterized thing. So the germ band just stops extending, there's no more extension, and no folds, right? No ectopic folds. So so this is sort of consistent with the idea that the ectopic folds are occurring from a compressive instability, and by cauterizing the germ band, you're not hitting the critical, you know, compressive stress to initiate instability, right? Now, then the question is, okay, well, so maybe the mitotic domains aren't enough on their own. Maybe all the compression is really coming from the germ band. So maybe there's the germ band on its own enough to do it. And so this is the double mutant that I had started talking about. I forgot the order that I had these slides in. So you do a double mutant, so you mess up the spike furrow and you mess up the mitotic domains. There's the germ band extension. No folds, right? So again, both of the two things that are generating compression in this sort of middle region of the tissue are not enough on their own to create the folds, right? But we know that they're enough together, right? And so that means now we have a nice way to qualitatively perform the fit that we need to fit for the bending rigidity, right? So now we know that we want the sort of parameter regime that we want in the model for the effective, dimensionless bending rigidity is the one where you generate, one or maybe sometimes two folds when you have the germ band and the mitotic domains on, and where you don't generate folds when the germ band only is on, or the mitotic domains only are on. And there indeed is a parameter regime where that happens, right? And so, we don't know exactly what the value is in there, but it's a relatively small window, and that is the window that's consistent with the physiological behavior for the model. And that's the only fit, right? So it's a one parameter fit. Everything else is taken either, by making a symmetric, choosing a symmetry, up front, or taken from measurements, all the other parameters. Yeah.
So I guess I don't understand. So if I look at the figure, at the germ band only, there is a parameter domain where you do get these things, right? In the middle panel and then down.
Yeah. So this is no good. So this is the model, right? And so the more down you go, the cheaper bend energy the bend modulus is. So it's easier to bend or harder to bend on the top row and easier to bend on the bottom row. And so now we know in the real system, we want to see folds on this column and not folds on this column or this column, right? And that lets us set the bending rigidity of the model that we would predict is a good match for the real system. So one way to say this is that this fit gives us a prediction. If people were actually able to measure moduli in a tissue, which is something that can't really be done at the current time. But if you can eventually, once one can eventually measure those moduli, then we have a clean prediction for what they're going to be, because that's the only regime that matches the physiological behavior. Does that make sense?
Yeah, well, it feels a bit like a circle.
How so? It's a fit, right? Like it's.
Yeah, so if it claims you need them both, then there is some domain where you don't need them both, right? So I'm a bit lost.
No. The claim is that you need them both, and that each one individually is not enough, right? So there's the data that shows that you get ectopic folds when you turn off the sphalic furrow, and you have the extension of the germ band and the mitotic domains, right? That was the first ectopic fold data I showed you. And then on the last couple slides, we had these examples, right? So like, are the mitotic domains alone sufficient to drive ectopic folding, right? We cauterize the germ band extension, so then we only have mitotic domains, and then there's no folding, right? So the mitotic domains alone don't make folds. And then we do the same thing for, is the germ band alone enough, right? And we do this by having a double mutant that messes up the mitotic domains, right? And then again, this is the double mutant down here. And then again, there's no folds. Okay? No folds. So yeah, so the idea is we have these three pieces of information from the data. And then we use those three pieces of information to sort of qualitatively fit, right, the regime of the one parameter that is a free fitting parameter in the model. Okay?
Which is a ratio, right?
Yes, the ratio of the bending
That's the ratio of
the stretching rigidity with an R squared data.
Yeah, but the prediction would be like you can say something about the relative strength of these two effects.
Right. Yeah, we can't make a prediction about the precise value of the bending rigidity or the stretching rigidity, but the prediction will be about the ratio of the two. That's a good clarification. Yeah. Question?
What's the time scale at which these movies happen?
That's another good question. A couple hours. if I recall correctly.
Okay.
So they're, on developmental timescales, it's pretty quick, quite quick, but you know, it's not on like an elastic response timescale, right? It's a couple hours. Okay, any other questions? Okay. Okay, and so now that we now have the model fit, we can go back and put the cephalic furrow into the model. So far in the model, we haven't had a sort of a programmed cephalic furrow. So now we can put one in, right, by generating, having a preferred curvature over a certain region of the model, which will drive, and this is the cells that are pulling down, you know, that have that special double gene expression. And so if you do that, then you basically create, you know, what we think of as like like a compressive sink, right? So that all the compressional energy just goes into the cephalic furrow that you programmed, and you don't get folding all over the place, right? So if you don't have the furrow, where the folds occur, you can have more than one, and they can occur sort of almost anywhere over a broad region of the tissue. Whereas if you program the fold, you get very, very cleanly, you know, the fold in the correct location, and almost always only one. There was this one, a very few cases where we got a very, very tiny little ectopic fold occurred just to the head side of the furrow, and we actually were concerned about that, and maybe we were running the wrong parameters. You know, we went to Pavel and Bruno and asked them about it, and they said, oh yeah, actually, in the wild type, you know, maybe, you know, 5 to 10% of the time, there's a tiny ectopic fold on that side also. So great. So that also, even though we thought that was a problem, that actually also even is a reflection of the real system. Okay, and so the idea, right, to sum up this part, right, the idea is that the cephalic furrow is playing a mechanical role, right, as a temporary sink for compressive stresses, right? And so why, oops. You know, essentially it's there to anticipate the gastrulation movements and the mitotic domains. And what it really is doing, right, from an evolutionary point of view, is it allows the fly, the Drosophila, specifically, Drosophila melanogaster, whatever, Drosophila, like the model animal Drosophila. It allows it to do gastrulation and germ band extension and the mitotic domains at the same time. And guess what? Not all flies do that. Right, and so we were doing this work, and we sort of ended up realizing that there was another group in Heidelberg that was working on the evolutionary side of the similar story, and so the reason here is that it allows you to speed up development significantly by not having to do these things. You can do them in parallel instead of in series by having this cephalic furrow. The flies that do them in series don't make a cephalic furrow. Surprise, surprise. And actually, there's another fly that does them in, there's a couple other flies like this Drosophila that does them in parallel and makes a cephalic furrow. Most flies do them in series and take longer to develop. And there's another small number of flies that do them in parallel, but instead of making a cephalic furrow, they do this funny thing where in the mitotic domains, they divide into the yolk. And the cells stay, the divided cells stay down in the yolk until the germ band is done extending, and then they come up, right? So they have a different solution, right, to speed up development than the Drosophila found, which is quite cool, right? So there's different ways to solve this problem. You know, you can either just take longer and then sort of be more susceptible to predation or whatever else, or you could speed up, and in order to speed up, you need to have this sort of extra genes and extra regulation to either do this or to do another thing. So there's this really cool, you know, evolutionary mechanical instability story happening sort of under the hood here. Okay, and so we have 10 minutes. We're going to shift to vertex model. So now we're going to go to real mechanical networks. So that was the sort of 1D simplified warm up. Any questions about the cephalic furrow story? That's it's on the archive, that story at the moment. So it's still, you know, making its way through the publication process, but you can read it. You can read the draft you can read the preprint on the archive. And so yeah, the 10 minutes until lunch, I would just try to quickly do the vertex model on the board, the intro, and then we'll come back to the slides after lunch with the story on the more full-blown network. Yes.
Can you stop sharing?
Ah, right. Yes. We do
Just
move
definitely have
the window. move the movie camera, whatever. How do I get? How do I hid the panel, the control panel, and now I don't know how to get it back. Let's start sharing. Oh, here we go. Yeah, thank you. Yep. Okay, so in many of those images I showed of epithelia sort of seen from the top down, right? And they really look like a polygonal island, right? They look like in the surface of the Drosophila embryo or in sort of some of the other images I showed. And so the idea of the vertex model, right, is to take color, is to take that sort of impression seriously, right, and model a tissue, the epithelial tissue, or at least the apical side of the epithelial tissue, as as a polygonal tiling with degrees of freedom given by the vertices of the right? And now that's simplifying things a little bit, but we'll come to that in a second. So, so you have this configuration where you have a network, right? So you have a bunch of vertices, you have edges, the set of lambda ij's, and then we have cell identities alpha, right? So you have areas, edges, and vertices. Okay. And the vertex model, you write down a work function or energy function for it. There's a couple different choices that people make, but one of the typical ways to write it down is to write it down like this. So summing over all the areas. And then summing over all neighbors. And then summing over, again, all areas. Like this. Okay, now what do all the symbols mean? Okay, so this is the area stiffness, right? So this is sort of the modulus of changes in, if it resists changes in area of the cells. This is the current cell area for cell alpha. And this is the preferred cell area for cell alpha. Okay. Now over here, right, we have the line tension, right, on the junction, the junctional line connecting, IJ, this sort of lambda, this corresponds to edge lambda IJ. And then this L is the length of lambda IJ. Okay. And then we have the perimeter stiffness. So this is a perimeter minimizing term here, and then this capital L is just the cell perimeter of cell alpha, current cell perimeter. Okay.
What is the difference between cell area and peripheral cell area?
So the cell area is the current cell area that the cell has, and the preferred cell area is the cell area that it wants to have. Right? So when it's different, so it's preferred. So when they're the same, right, this minus this is zero, and this whole term goes to zero, so it doesn't contribute to the energy. But when it's different, right, it contributes to the energy like the difference squared times the modulus, like just like a spring, right? The energy goes like kx squared, right? So it's like a springy, elastic energy associated to area deviations. And this is also an elastic energy associated to the perimeter being different to zero, right? So one usually assumes that the preferred perimeter is zero. And then there's an energy coming from how much tension you have on the bonds, right? Okay, and now if you look at this and you think, okay, so like a typical tissue with when one wants the model is going to have, order 100 or 1,000 vertices and a similar number of edges, and maybe, if it's 1,000 edges and vertices, then it'll have something like 200-ish cells, right? Every single one of those cells has its own area stiffness and its own preferred area and its own perimeter stiffness, and every one of those edges, might have its own line tension or length, right? Like it seems like this is like wildly over. There's a danger of a wild overfitting going on here, right? So in practice, so this is the general form of the work function for the vertex model. In practice, what people do, if you take, maybe we'll take blue. So now in practice, I'm going to cross off stuff. So in practice, this doesn't depend on alpha, right? So the area stiffness is the same for every cell in the tissue, right? The moduli. And also the preferred cell area is the same for every cell in the tissue, right? So that's a cell that maybe you go by cell type. Maybe there's 2 cell types and you have 2 different ones. But usually you just have one kind of cell where they have the same stiffness and the same preferred cell area. And similarly, you get rid of this degree of freedom as well, right? So you cross off the fact that every single cell can have different values of the stiffnesses and the preferred areas. And then it's not as bad, right? So then these are the actual parameters you now have to fix are really, comes down to, just one area stiffness, one perimeter stiffness, and one preferred cell area. Right, so then it's much more, you don't have this wild overfitting problem potentially if you're trying to mimic a tissue. Okay, and then so now if that's the energy, right, like how do you want to actually evolve this thing? Right, so then you can calculate the forces on every vertex. Notice that like there's this area term. It is possible to write an area as a function of the vertices that surround it, so long as you need to know the network, right? You need to know who is whose neighbor, and you need to know for any given cell alpha, you need to know which vertices they are, right? That are its bounding vertices. So you can rewrite this thing. It looks horrible, like it's a mess, but you can rewrite it purely in terms of the vertex locations, right? With the same energy, with an area energy and a tension energy, right? And so then, because you can write it purely in terms of the vertex locations, you can then calculate the force on each vertex, which is going to be equal to just the spatial gradients of the energy. Right, so it's just going to, you want to just go down, it's kind of we're making a quasi-static approximation here. There's not like, some kind of momentum baked into the motion. We're just going to go down the energy gradients. And then when as the tissue configuration that you're at is stable, if the force on every vertex is 0 for all vertices, and if the equivalent of the Jacobian rate is negative that. Okay, for all I. Right, so then, and then you evolve the thing until you end up in some stable. You can, you know, lots of people sort of inject noise in different ways. You don't have to, or you can just get stable stuff. Okay, but now to capture actual tissue mechanics, what I've told you so far is actually, it looks complicated and it looks kind of nice, but it's actually super boring, right? Because all that's going to happen is that you put down, you know, some additional configuration, and it's going to minimize the energy elastically, and then it's just going to stop, and that's it, right? Nothing interesting is going to happen, right? And the reason why that is, right, is a little bit hidden. I said it at one point, but it's worth like really emphasizing this point. That energy, despite the fact that I didn't, you know, it's not written there explicitly, it really depends on the neighbor interaction network that gives you like who is sharing the edge with who, and which cell has which, you know, which neighbors, which cell has, you know, what vertices comprise each cell. Right, that network really matters, right, for the energy. And if there's a change in that network, right, if suddenly like one of these cells sort of delaminates and disappears, for example, or the cells change neighbors, which happens all the time in a relationship, right? If the cells change neighbors, right, you have something that looks like this. You have some quartet of cells that looks like this, you know, embedded in the tissue. And then these two can sort of, if this edge gets small enough, then these two can become two neighbors and drive apart these two as being neighbors. Right, you can get transitions like this. And in fact, this is one of the sort of minimal transitions that occur in a topological, in a 2D topological network like this, right? In a spatial network like this. So this is sometimes referred to as a T1 event, or a T1 transition. T1 event, T1 transition. Sometimes it's just called a neighbor change.
These are, I mean, you say occur means you observe them.
Yeah, you see them happening in the real tissue.
Exactly, because in this,
exactly,
the setup of
exactly.
this model now, there is no dynamics.
This doesn't have, well, there's this relaxation dynamics, right?
But that is not a,
but you're not going to get this.
it's not a real, where's the relaxation? I mean, it's not a real dynamics, it's a minimization.
It's quasi-static relaxation dynamics. Right, it's not real dynamics, but it's you know, if you're working under the quasi-static approximation, it's reasonable. But you don't get this, right? You don't get anything interesting. And so what people then do to make this an interesting model.
Sorry, I don't understand. This dynamics that you have written down there is a minimization.
I'm sorry, yes.
And so I think there is no.
You're right.
Because you don't have a time scale. You don't have any quantity with a time scale in your system at the moment.
That's correct. Yeah.
I mean, it
But
could
you
be
can,
that
right, you can take this, right, and write down a quasi-static relaxation along these forces with an arbitrarily introduced time scale.
Yeah, exactly. But then you do some kind of
Yeah. That's exactly right. Yeah, I'm sorry. Yeah, I forgot to write that down. Yeah, you're right.
I'm not an expert in this, so I'm interesting to think.
But the point is that people do that sometimes, but it's not that interesting because of this, right? Because you actually need these to have anything interesting happen. And the timescale that these occur on is the timescale that matters. And then, so what people actually do is you come up with some other rule where you make these happen by hand, or you have a rule that says, okay, if an edge gets short enough with some cutoff, then I like do like almost like a Monte Carlo move, and I like try to flip it and look what happens to the energy. And then with some Boltzmann factor, I take it or not. You know, and then those things are occurring on some time scale. Those possible swaps are occurring either in a driven way on some time scale that you introduce. And then you assume that that's time scale separated from the relaxation. relaxation. So you just do the relaxation, don't worry about the time, the dynamics of the relaxation. So in practice, that's what really happens. There are cases where people do put a time scale on this and worry about the elastic part, but most of the time people will care about putting a time scale on this or another topological change, right? And so the point is that there's all these sort of energetic details of the vertex model, and they are what makes the vertex model what it is. But to make the vertex model do anything interesting, right? Like that's just the background. What actually matters is what rules did you choose for the topological changes and how do you handle topological transitions? And I would sort of close, you know, when you go have lunch on maybe much more broader philosophical point that anytime you have a continuum model, in physics, in anything, right, in any discipline at all, right, continuum models don't play nice with topological changes or transitions. And the reason why that is if you remember all the way back, and I think the very second slide of the first day, right, you know, there's geometry and topology are these two aspects of shape. And geometry is about continuous changes. And so if you have a continuous model, it's telling you about geometry. And topology, everything about topology changes discontinuously. I either am or am not a neighbor with, you know, vertex J. And, you know, at one time I am, and at another time I'm not, and at no time am I half a neighbor. That doesn't mean anything, right? You have discontinuous changes that you can't capture with smooth derivatives and continuum models. And so if you have any kind of continuum model, and then topology changes matter for the thing you're modeling, then you need to handle it somehow separately in some ad hoc way. And that's what people do. But I think I would point to that as actually being a really, really interesting, deep problem in the way we do modeling, you know, across all the sciences right now. So there's no coherent way to combine dynamical models or models that capture change in continuous senses and discrete senses at the same time. It's all, you know, there's ways to do it on a case-by-case basis, but there's no unified vision of it. And I think that's kind of crazy. And let's have lunch. Any questions? Yeah.
What is the time scale of T1 transition generally in terms of scale?
Oh, it depends on the tissue. It depends on the behavior. You know, sometimes in some tissues, the rate, you know, they don't exchange neighbors very often at all. It's quite quiescent. Sometimes there's a quite high rate relative to the more genetic rate. You know, where you see changes happening on the order of minutes, let's say. So it's quite variable.
Carl, can I ask you a question?
And we'll come to an example after lunch of a tissue that has these T1s going on and they do something.
Carl, can you hear me? Can I ask a question?
Okay, so I guess, oh, is there a question online?
Yeah, well, is Deb here?
You're muted maybe, or?
No, well, I'm not muted, but I'm not sure
We
how.
can't hear you, hold on. Oh, I logged in without turning on the audio on my computer, so.
Here it says conference.
Deb's not actually muted, I don't know.
There is a Teams.
Oh, maybe I have to join, but it shouldn't matter, right? You should be able to do it from your end.
Yeah, I'm unmuted here.
Oh, here we go. Wait a second. Go ahead.
Yeah, can you hear me now?
Yeah, not well, but a little.
I can type the question if that's useful.
Yeah, I think
although
it's probably
good.
easier. I think only I can hear it. It's really soft for some reason.
Although it's more like a comment than a question. What you just made a remark on, that there is no way to combine the discrete and the continuous changes. That's not entirely true. And this is exactly what, I mean, let's say at an overarching level, that's basically what I nowadays do, is that the discrete changes are mapped by the changes in the networks, because networks by construction are discrete. And then the processes that, because, you know, if you don't have a connection between A and B, then there is no processes that can play over that connection. And so connections you can still model continuously while the network is discrete. So you can do it. And these are the temporal networks as we have been talking about in the last 2 days.
Yeah, so just for everybody else, what Deb is saying is that one can, you know, if you're thinking about dynamics playing out over a network, for example, you can do, this is a comment on what I said at the very end about not being able to combine the sort of discrete and continuous changes. So, Deb was arguing that, you can do it, because all the processes just go to zero, and don't have a link anymore. And I agree with that. And I did not mean to say that one can never do it. You can do it actually in individual cases, you can almost always do it. But what I would argue is not present is a more general formalism that allows you to do it across multiple different classes of cases in the same way. So I think, for example, that what you said wouldn't really work for other kinds of, for the transition, for example, like of a membrane, right? A membrane budding or rescission transition. For example, one typically, in order to get like all the scalars right for that, you have to do multi-scale simulations, and you have to like stop and then zoom in and do molecular stuff and back out.
Yeah.
So it's a different solution, right?
Yeah.
That people use.
Yeah, I can tell you that there is no one size fits all. The remark is kind of an overarching remark, and it is very hard. It is really very hard.
Yeah, fully agreed.